The process of cell division, both meiosis and mitosis, transmits chromosomes that are morphologically intact and unchanged from one generation to another. At times, however, errors occur that result in the modification of one or more chromosome within a cell, resulting in cells and individuals with abnormal structure or numbers of chromosomes. Such visible chromosomal abnormalities have been found to occur in about 6 or 7 of every 1000 live births. A study of chromosomal abnormalities is important as these are associated with problems like infertility, miscarriages and several kinds of birth defects, delayed puberty, hermaphroditism and cancer. You will study chromosomal abnormalities due to change in their structure in this module. Such changes are also referred as chromosomal aberrations. Aberrations of structure refer to the changes in the part of the individual chromosome rather than whole chromosomes or set of chromosomes in a genome. There are 4 types of structural alteration: deletion and duplication, which involve a change in the amount of genetic material on a chromosome; inversions, that involve a change in arrangement of the chromosomal segment; and translocation, which involve a change in the location of a chromosomal segment. You would study these 4 basic types of aberrations and their effects in this module. Deletions. A deletion or deficiency is a chromosomal change resulting in the loss of a segment of the genetic material and the genetic information contained therein. A deletion may be as small as a nucleotide, though such a short segment cannot be seen cytologically, or it may be a large chromosome segment. Deletions may be caused by agents such as heat, radiation, viruses, chemicals, drugs, or due to errors in recombination. The deletion where the chromosome breaks off near the end, it is called a terminal deletion. If there are 2 breaks within a chromosome arm, the segment is lost. It is called an interstitial deletion. The chromosome segment that lacks a centromere, known as the acentric fragment, will be unable to attach to the spindle fiber, and consequently it will not be included in the nucleus of either of the daughter cell. Such fragments are ultimately degraded and are lost from the genome. Genetically, deletions are distinguished from gene mutations. In the case of mutations, the genetic material may back mutate, but in deletions the loss is permanent, as the genetic material is no longer available for further change. Cytologically, deleted chromosome sections can be detected in prophase of meiosis first. If cell is heterozygous for deletion, that is, it has a deficient and a normal chromosome, then during synapsis the chromosome pair precisely band for band along all the homologous regions. In the deficient region, however, the normal chromosome will not have any region with which it can pair. Therefore, a small unpaired chromosome segment buckles out to the side. It is therefore known as buckling effect. Such a configuration is called a deficiency loop or compensation loop and can be observed under the light microscope. The consequences of deletion depend on the genes or parts of gene that have been removed. In diploid organisms, the effects may be lessened by the presence of another set of the deleted gene in the homologous chromosome. However, if the homologue contains recessive genes with deleterious effects, then the consequences can be severe. We therefore know mostly about those deletions that cause a detectable phenotypic change. With the use of high resolution banding techniques, a growing number of interstitial deletions have been found to be associated with human diseases of previously unknown causes. Deletions have also been identified in the X and Y chromosomes and most of the autosomes. Virtually all deletions of euchromatic band result in mentally retarded babies having multiple congenital anomalies. Note that several of these deletions listed in the table cause cancer. Deficiencies occur in humans as well as in other organisms. We shall discuss two examples that result due to such chromosomal aberrations in humans. Cry-du-chat syndrome, a well-known disorder associated with the deletion in humans, is Cry-du-chat syndrome, described by Lejeune and colleagues in 1963. The children suffering from this disorder have a characteristic high-pitched plaintive cry very similar to a cat in distress. Accordingly, this disorder was called in French the Cry-du-chat, cry of the cat syndrome. This syndrome is associated with a malformation of the larynx. The cry, facial appearance and other abnormal physical features and an IQ, intelligence quotient that tells us about the level of mental capacity, usually below 20, are so characteristic of this deletion that the diagnosis can often be made prior to karyotype analysis. Other non-specific malformations and dysfunctions involving the brain, heart, eyes, kidneys and skeleton may also occur. Nevertheless, many affected individuals attain adulthood. The frequency of this defect is about 1 in 50,000 live births. Twice as many live born females are affected than males. This disorder results due to a deletion of a segment of the short arm of chromosome 5, precisely the parts of the band 14 and 15 and the region in between. The karyotype is thus represented as 5 del P14 P15. In most cases, the deletion arises during gametogenesis in one parent or the other. Both parents of the affected children, however, present normal karyotypes. No parental age effect is discernible. Myelocytic leukemia. Patients with chronic myelocytic leukemia carry a short chromosome named the Philadelphia chromosome PH1 after the city where the first case was discovered. In all the bone marrow cells and cells originating in the bone marrow, that is neutrophils, eosinophils, normoblasts and megakaryocytes. During remission of PH1 persists in bone marrow and often in cells derived from there. Detailed cytological study discloses PH1 to be a number 22 chromosome that has lost most of the distal part of its long arm 22q. This deleted part of autosome 22 is often seen to be attached, translocated to one of the other autosomes, preferentially to the distal end of 9q, and reciprocally a small fragment of 9q is translocated to 22q. Duplications. When any part of the genetic material, a single locus or a large piece of a chromosome is present more than once in a genome, it is called duplication. Such chromosomes, that is one with duplicated portion and the other one normal, produce compensation loops at the time of synapses. Similar to those described for chromosome deletions, duplications arise as a result of unequal crossing over between synapsed chromosomes during meiosis or through a replication error prior to meiosis. In the former case, both the duplication and deficiency are produced. When present on the same chromosome, it may be present in a tandem configuration, that is AB, CB, CDE dot FGH, that is adjacent to one another and in the same chromosome. Sometimes, however, the order of the genes in the duplicated segment are reverse of the original. For example, AB, CC, BDE dot FGH and is known as reverse tandem configuration. And when the duplicated segments are tandemly arranged at the end of the chromosome, it is called terminal tandem duplication. for example, AB, CDE dot FGH. Small duplications do not usually have as drastic effect as deficiencies of comparable size. Thus, a small duplication is rarely lethal even when homozygous, whereas small deficiencies may be lethal even in heterozygous condition. However, sometimes duplications can be pathological. The first duplication to receive critical study was the bar eye variant in Drosophila melanogaster. The wild type eye is oval shaped and the bar eye phenotype is characterized by a narrower oblong bar shaped eye with fewer facets. The classical studies of bridges showed this trait to be associated with the duplication of a segment of the X chromosome called section 16A as observed in salivary gland chromosome. Each added section 16A intensifies the bar phenotype. However, the narrowing effect is greater if the duplicated segments are on the same chromosome. There are very few reports of duplication in human beings, as small duplications that have little or no phenotypic effect largely escape notice. One example of duplication is DUP9P. The affected persons are now mentally retarded, having a good life expectancy. Inversions. Chromosomal inversions, another category of chromosomal structural variations, involve two breaks either in one arm of a chromosome or one break on either side of the centromere. Following the breaks, the chromosome fragments between the break points rotates through an angle of 180 and is reinserted into the chromosome in such a way that the genes are in reversed order. For example, if a chromosome contained the following gene order A, B, C, D, E, F and a break occurred between genes A and B and between D and E, after rotation and reinsertion, the chromosome would read A, D, C, B, E, F. Figure demonstrated how an inversion might arise. In this, the newly created sticky ends are brought close together and rejoined. The inverted segment may be short or quite long and may or may not include the centromere. If the centromere is not part of the rearranged chromosome segment, the inversion is said to be paracentric. If the centromere is a part of the inverted segment, the term pericentric describes the inversion. Although the gene sequences has been reversed in the paracentric inversion, the ratio of arm lengths extending from the centromere is unchanged. In contrast, some pericentric inversions create chromosomes with arms of different lengths than those of the non-inverted chromosome. Thus, the arm ratio is often changed when a pericentric inversion is produced. The change in the arm lengths may sometimes be detected during the metaphase stage of mitotic or meiotic divisions. Pericentric inversions are responsible for some of the changes in chromosome configuration that take place during evolution. For example, the human chromosome 17 is acrocentric while the corresponding chimpanzee chromosome is metacentric. Inversions in general do not result in a loss or gain of genetic material, although there can be phenotypic consequences when inversions occur within genes or within regions that control gene expression. Genes, when placed into a new setting, as happens in inversions, may lose their regulatory controls or alter the activity of genes in the area. In individuals that are homozygous for a chromosomal inversion, the linkage order of genes becomes changed. A homozygote for the A, D, C, B, E, F chromosomal arrangement will show a closely linked to D rather than to B, as in the original sequence A, B, C, D, E, F, and E closely linked to B rather than to D. In inversion heterozygotes, synapsis of homologous chromosomes requires the formation of loops containing the inverted segment. Heterozygous inversions can be recognized by the presence of such loops in the preparation of cells at the packaging stage of meiosis. Heterozygous inversions can also be identified in genetic experiments, since recombination is reduced significantly or suppressed. In the figure, the effects of a single crossover in the inversion loop of an individual heterozygous for a paracentric inversion are shown. During the first meiotic metaphase, the two centromeres migrate to the opposite poles of the cell. As a result of the crossover between genes B and C in the inversion loop, one recombinant chromatid becomes stretched across the cell as the two centromeres begin to migrate, forming a dicentric bridge. As the two centromeres continue to migrate to the opposite poles in the cell, the dicentric bridge under physical tension breaks randomly. The other recombinant product of a crossover event in chromosome without centromere, an acentric fragment, is unable to continue through meiosis and is lost. That is, it is not found in the resultant gametes. In the second meiotic division, the centromeres get divided and the chromosomes are segregated to the 4 gametes. 2 of the gametes have complete sets of genes and are viable. The gametes with normal order of genes A, B, C, D, E and the gametes with the inverted segment A, B, D, C, E. The other 2 gametes are unviable, since they both are deficient for many of the genes of the chromosome. In general, the only gametes produced that can give rise to viable progeny are those containing the chromosomes that were not involved in the crossover event. The effect of crossing over in an individual heterozygous for a pericentric inversions are shown in the figure. Of the 4 chromosomes resulting from the meiotic divisions, 2 form viable gametes with the non-recombinant chromosome A, B, C, D normal and A, C, B, D inversion, and 2 recombinant gametes that are unviable, each as a result of the deletion of some genes and the duplication of other genes. Usually, only the gametes containing the 2 complete chromosomes can produce viable progeny, and normally therefore, no genetic recombination is found in the progeny of heterozygote for either pericentric or paracentric inversions. It should be noted that not all crossover events within an inversion loop lead to unviable recombinants. One exception occurs if a double crossover takes place in the inversion loop and the same 2 chromatids are involved in each crossover. A second exception occurs when the duplicated and deleted segments of the recombinant chromatids do not affect gene expression and hence viability to a significant degree, as when the chromosome segments involved are very small. Translocations. This is the 4th kind of structural alterations in the chromosome. It is also called a transposition and involves the movement of segment of a chromosome to a new place in the genome. Translocation may occur within a single chromosome, intrachromosomal, or between non-homologous chromosome, interchromosomal. In the interchromosomal translocation, there are 2 possibilities. One, there is transfer of one chromosome segment to another chromosome and it is known as non-reciprocal translocation. In the second case, there is reciprocal exchange of segments between the non-homologous chromosomes and it is called a reciprocal translocation. By definition, a reciprocal translocation is balanced if there is no loss of genetic material or the DNA, and the unbalanced if there is loss of genetic material. The consequences of the translocation to the production of meiotic products depend on the type of translocation involved. In many cases, some of the gametes produced have duplications and or deletions and hence are unviable. We concentrate here on reciprocal translocations, since they are the most frequent and are found in 1 in 500 live births in humans. In homozygotes, for a reciprocal translocation, meiosis proceeds normally, since all chromosome pairs can synapse to produce bivalents, whereas in heterozygotes for reciprocal translocations, unorthodox synapsis occurs during meiotic prophase first. This results in the formation of cross-like configurations owing to the requirements of pairing between homologous chromosome parts. Instead of bivalents, that is pairs of synapsed homologous chromosomes, there appear quadrivalents consisting of 4 associated chromosomes, each chromosome being partially homologous to 2 other chromosomes in the group. Segregation at meiotic anaphase may occur in 3 different ways, producing 6 types of gametes. Of these gametes formed, only the 2 in the figure which result from alternate segregation, that is the 2 chromosomes derived from the same parent go to the same pole, contain all the chromosomal parts, that too only once, and no duplications or deletions occur. All the other gametes, the remaining 4 type seen in the figure, which result from adjacent segregation, in which the 2 chromosomes going to the same pole are derived one from each parent, have some chromosome segments duplicated and some deleted. These therefore cannot form normal progeny. Translocation heterozygotes are semi-sterile owing to the production of abnormal gametes. In plants, pollen grains with duplicated or deleted chromosome segments are usually aborted. Animal gametes with duplicated or deleted chromosome parts may function, but the zygotes formed by such gametes usually die. However, if the duplicated and deleted chromosome segments are small, the gametes may function and produce viable offspring. In conclusion, most structural abnormalities in chromosomes are of one of 4 types: duplications, deletions, inversions and translocations. The basic mechanism of structural aberration consists of breaking and rejoining of chromosomes. Linear linkage relation among genes in chromosomes are altered by structural aberrations.